Hello everyone and welcome to Meds.Concepts. Today, we are diving deep into one of the most critical topics in microbiology, sterilization and disinfection. We'll cover the core principles, advanced methods, and the best practices to ensure safety and validation in the lab. Why do sterilization and disinfection matter? It's more than just cleaning. It's about safety and accuracy. Here's the impact. Aseptic control. It prevents cross-transfer and ensures experimental validity. Contamination prevention. It lowers false results by keeping air and surfaces microbe-free. Safety. It protects lab staff and patients from infectious risks. Sterility confidence. It verifies that every item meets safety thresholds before use. Modern relevance. Essential for handling resistant organisms in today's automated labs. The bottom line, these practices are the foundation of trust and scientific progress. First, let's understand the basics. Sterilization isn't just cleaning, it's about the complete inactivation of all microbial life, including highly resistant bacterial spores and prion. We must also consider the bioburden, which is the number of microorganisms present before the process, and choose a method with a broad activity spectrum. Moving on to physical methods. Moist heat using an autoclave is our gold standard for rapidly coagulating proteins. For moisture-sensitive items, we use dry heat. We also utilize membrane filtration for heat-sensitive liquids and radiation for deep penetration and surface decontamination. When heat isn't an option, we turn to chemical agents. From alcohols and chlorine to advanced aldehydes, the choice depends on the material's compatibility and the required level of disinfection. Remember, the right concentration is key to effectiveness. Looking towards the future, we are adopting low-temperature technologies like hydrogen peroxide plasma and ethylene oxide. These are essential for modern, heat-sensitive medical devices, ensuring they are sterile without being damaged. Validation and quality. How do we know it worked? We use the sterility assurance level of 1 in a million as our benchmark. This is verified using a hierarchy of indicators, from physical parameters and chemical strips to the ultimate proof, biological indicators. Success depends on discipline. This means correct PPE, thorough pre-cleaning to remove organic debris, and ensuring the right contact time. Proper packaging and meticulous documentation are what prevent avoidable failures in the field. In conclusion, always select the right method for the right risk. Continuous improvement and rigorous quality control turn a simple procedure into a reliable life-saving system. Always match the decontamination intensity to the microbial burden. Thank you for watching. If you found this guide helpful, please follow and subscribe to Meds.Concepts to stay connected for more medical and microbiological insights. See you in the next video.